B2.3 or cell specialization. So, alright, so what is cell specialization? Well, basically, it's when a cell discovers or starts to do something very specific, so something very special, and that starts off quite early. So, obviously, from our previous topic, we know that a cell starts off as a little gamete, so we're gonna say gamete, then that, so two gametes make a zygote. And this is actually the first example of a full cell because it has your 46 chromosomes, and we actually, all humans, like all of our cells other than our sperm cells and our egg cells, are going to be just zygotes, right? But within the zygote, there's a lot of categories of cells. So you have your stem cells, and then you have your specialized cells that all have very certain functions. Now, what we're going to talk about today is stem cells and expand on that topic, right? So let's actually start to talk about this. So the first step is that zygote is gonna have we're gonna have a bundle of cells. So we're gonna have a zygote, and that's the very first step. So fertilization occurs, let's put that in green because fertilization in life. So fertilization occurs. Now, once we have a fertilized cell, which is a zygote, we very quickly have that multiply. Now, that multiplies and forms a blastocyst, and a blastocyst is actually a little bundle of cells, and these cells have infinite potential, so can become anything that they want, right? So just call them totipotent. So that's the actual terminology we use. So they can become anything. From a blastocyst, we're gonna actually move and we're gonna have a embryo. Now this is our main discerning step. At this point, things start to change, right? Now, in an embryo, these totipotent cells from the zygote become embryonic cells, and these cells are now going to be pluripotent. So, pluripotent, and the embryo, unlike the blastocyst, is gonna start forming many like little organs, and it's gonna form your head, your hair, everything kind of just starts forming. And the importance behind that is each of these cells has a certain function, and that certain function is actually decided in a very interesting manner. So let's actually talk about how that function is decided. So right now we're at embryos, and we're learning how those embryos allocate roles. Well, the best way to talk about this is through morphogens. Alright, now morphogens, like the name would suggest, just to remember, morph is to change or take form, and gen, you can pretty much say that however you want it, like the genetics take shape or the genes take shape, but in a sense that's what it is. It's the change or the taking form of the genetics. So morphogens actually start to get secreted from the developing embryo. So from embryotic cells, and that creates a concentration gradient. concentration gradient. Now, why am I telling you this? Well, mostly because it's pretty important. Morphogens are what we call transcription factors. So, transcription factors, which means they actually decide what gene is expressed. So the further away you stray from the embryonic cells, so the further away you go from the source, the lower the concentration is. And as you go down, you actually get a lowered concentration, so it starts to control for very specific transcription factors, and it codes for something more specific. Now, we know that at this point we have our pluripotent cells, but what we're getting here is actually a little bit different. We start to see multipotent cells. Let's actually write this in green. So multipotent. And these multipotent cells are actually what we find commonly in you and I in many places, because they allow that cell to differentiate into many types of cells within a category. So totipotent cells could have become any cell, and they become embryonic cells. Then you have your pluripotent cells, which can become a range of multipotent cells, and your multipotent cells, all of these are stem cells, by the way, and your multipotent stem cells, which can become just general cells. So they differentiate into regular cells. Now, let's actually look through this and see what we need to know specifically. So let's erase this and just quickly outline the three main types of cell. All right, so when you're conceived, during your big hoorah moment, we start off totipotent. And that is your zygote. So we're just gonna, boom, 46, your zygote. Then you actually go to pluripotent. Potent. And here, you're gonna be your embryo. So you're gonna, let's just draw you as like a really, call that a sac, embryo of sorts. Right? And you get your pluripotent stem cells here. So actually, let me actually just specify stem cells in this case. Then, from your pluripotent cells, you go down a step and you get your multipotent cells. Now we're going to erase stem cells and actually change the category, because I'm going to introduce a new concept, or a very old concept, which is just cells. So all of these guys up here are your stem cells. And this guy over here is it's just your cell, right? These guys can't change. They are locked to be who they have to be, and that's decided by morphogens, which, let's actually write that here, control gene expression. So remember, morphogens control gene expression, they morph your cells. All right, from this point on, let's actually take a closer look at cell niches. So we know that this is your adult stem cells. Adult stem cells. So let's take a closer look at that, right? So all right, multipotent. So what cells could be multipotent? Well, just very off the top of my head, everybody knows bone marrow. So bone marrow. Now that really differentiates into a wide variety of stem cells, but we have something slightly more niche. So these are called the niches, and that's going to be your hair cells. So hair follicle cells. And these cells are both, these are both just cell niches, yeah. So they found their place and they can differentiate into a range of regular cells. Bone marrow cells, additionally we can call these pretty much just things that form blood cells, are also called hematopoietic. Cells, I think. Let me just double check the spelling of that. So there we go. Cells. So these are your blood cells. All right, so we've now covered multipotent adult cells. So we're going to just write adult stem cells, and most of these are going to be multipotent. You're gonna, after a certain period of time, you're just gonna not have that many pluripotent cells because you don't need them anymore, right? They're not really doing much. And an interesting thing to think about is to actually make a fetus or to clone a baby or whatnot, you're going to need your original zygotic totipotent cell. And for that, you can't just make one because you can't reverse engineer this process. You're actually going to have to steal it or like take it from a baby. And that brings a lot of ethical problems in. We can talk about that later, but that's just an interesting, slightly disturbing thing to note. Now, we're going to talk a little bit about various size differences because there is a scale. So let's start off by just defining a quick scale. There we go. And the scale will help kind of put things into perspectives. So up here, you're gonna have your, say, your 2 micrometers. So like that. And at the very edge of the scale, we're gonna have 300 millimeters. So that's much longer because 1 mm is equal to 1000 which is micrometer. So here we have everybody's favorite swimmer, the sperm cell, right? So you got that, and you got your acrosome, right? So you get that at 2 micrometers. Then we're gonna go up to our red blood cells. So let's actually draw this to scale if we can. So a little bit like that. Then we have our red blood cells. So RBC, which is gonna be 8 micrometers. Then we move on slightly to just a cell, what everybody thinks of. So we go 12 to 17 micrometers. After that, we're gonna go on slightly. Oh, sorry. This is an interesting thing. So for sperm cells, we're actually talking about the width here, not the length. So width of a sperm cell. The length of a sperm cell is actually 55 micrometers. So you can say either one, but it would be good to define it. Then you go on to a human ova. I'm going to erase this because that. Go into a human egg cell. Like that. So about 100, I'd say. Yeah, so 100 so micrometers. Then nerves. So your nerve cell, little dendrites, axon terminal, myelin sheath, and that's about 300 micrometers. And then you got muscle cells, which is all the way at the end of this, very, very, very long. And we know that muscle cells are mononucleated, or well, they have multiple nucleus within a single cell, and that's because they kind of just join together. So we're going to draw multiple nucleus, that you get muscles, right? So this is how kind of grades upward. So we're going to erase that, and we're going to talk about a little bit about why things are different sizes. So obviously sperm cells have different parts to them, so it's a good thing to note this, because it can be asked, although not directly. So you have a sperm cell, right? So like that, this is 55, and this is about, say, 2 to 3 micrometers. And you have your egg cell, which is much larger, at a whopping 100 micrometers. Now, this sperm cell needs to fertilize, so let's green again. So this is going to have a 23 inside, and this is going to have a 23. They need to fertilize, and to do that, the sperm has an acrosome. So acrosome, enzyme filled, and those enzymes break down the outer layer, the outer membrane of the eggshell, and fertilize it. But that's just a little bit of interesting jargon. So we're going to erase that and move on to the next idea. So now we're going to have to talk about the movement of materials across a cell membrane, and the easiest way to do that is actually just draw it out. So this is still under specialization, but it's starting to talk about why certain cells have certain sizes, right? So we're gonna have a 1 by 1 cube, so just say 1 unit, right? The volume, right, V is equal to 1 to the power of 3, which is just equal to 1. The surface area is equal to, let's see, 1, 2, 3, 4, 5, 6. So it's going to be, this is just 1 to 1, so 1 squared times 6, which is equal to 6, right? So this is unit cubed, unit squared. And the idea behind this is that the larger the cell is, the less surface area to volume ratio that it would have. So here, the ratio here is 6 to 1, right? So surface area to volume is 6 to 1. But let's consider a different option here, where we have a little bit of a bigger cube. Accident, so B2.3. We have a little bit of a larger cube. Here we're gonna have this is 1 and 1, this is 1, this is 1, and this is 1. So this will be 2 times 2 times 2. So that's gonna be a volume equals 8, surface area equals 2, 4, 6, 24. And if we just divide this, we see that the ratio goes down from, well, the ratio is now going to be 24 to 8. And that's actually simplified down to 3 is to 1. So we've essentially just halved our ratio, and certain cells need for transport, right? So let's erase this. For transport to occur, cells need to be a certain size. So say this is a cell. We're gonna write it in red because this is very important. And there was a question on this that we had for membrane transport. Port SA to V is crucial. Why is that? Well, for energy to actually be produced, we need water to be entering our cells, we need oxygen to be entering our cells, and at the same time we need stuff to be getting pushed out of our cells. If the cell is extremely large, right, so this is an extremely large cell, this is say a small cell, we can do this, we can do this, we can do this, right? The rate at which, so the rate at which the compounds can move in and out is actually going to be significantly so per unit volume than here. So we actually will have so much volume to fill within the cell that we're not able to actually make use of all the metabolic processes, and it just kind of causes our body to cease functioning. And that's why a lot of cells kind of cap out. You don't get cells that are massive or like really, really tiny. You do have cells that are tiny, but not too many. So there's like a little, the sweet spot of cells, which is about 12 to 17 micrometers. And that's just because metabolic processes within the cell are the most optimal at that range. Erase this. And now we're going to get into some of the HL content. So in the HL content, what we just spoke about is going to be covered more heavily, and I will talk about that in the next video.